Yes.
Actually, last night I checked one of the check again your slide.
Yeah.
And one of the slide mentioning the UV damage to the DNA in UV actually activate the check one.
Yeah.
And it is inhibit the some sort of like V1.
We want kinase, yes.
Yeah. My question is that when I look up the literature, it say that they only phosphorylate the V1,
Mm-hmm.
but they didn't say it's inhibit V1.
Inhibit, okay. So the TRK1 doesn't have an impact to phosphorate these.
Yeah.
And somehow the literature maybe just look it up more.
Okay.
So the capability of the check to regulate the V1 kinase is indeed by phosphorylation and will reactivate the V1 kinase activity. At the same time, the check one will be able to phosphorate on CDC25 at the same time.
Okay.
Yeah.
Okay.
Good.
Yeah.
Yeah. Question here.
So I want to mention about the protein 1433.
Yes.
It has been important role between yeast and EQ3. And you say in some cancer like breast, lung, is downregulation or loss expression.
Right.
So is there anybody try to find out the reason or the mechanism of this? Because I think if we find out the reason or the mechanism, we can make it re-expression and make the cancer maybe in differentiation.
Correct. Yeah, it's a very good thought. Actually, it's very been found apart from our own lab study, apart from other lab study. So the downregulation of 1433 sigma. So he was asking what's the mechanism behind 1433 sigma's downregulation in many type of cancer. It was found that actually the promoter region of 1433 sigma is highly methylated and thereby causing the downregulation of 1433 gene expression. So that's one mechanism. The second mechanism, I think we also talk about another E3 ligase called EFP. Okay, so EFP is the E3 ligase involved in 1433 downregulation. Although EFP's overexpression in cancer has not been defined. Okay, recently in my lab, we have another discovery is showing that another E3 ligase is able to downregulate 1433 sigma, which is COP1. Okay? The COP1 E3 ligase is able to cause the downregulation of sigma by enhancing sigma's ubiquitination and degradation. And the COP1 is overexpressed in many types of cancer. So the mechanism could be coming from transcriptional level regulation due to the promoter methylation, and the other mechanism is due to the enhanced degradation of 1433 sigma. Could be due to the E3 overexpression of sigma, such as COP1. Okay, good thought. Okay. Any other question? Yes.
Yesterday you talked a lot about the protein 1433 sigma. So I wonder the other protein in this family, like 1433 ampere. Yeah.
Okay.
They have same. They have a same characteristic like the sigma one.
Okay. So he was asking whether other isoform of 1433 family members share the same type of regulation like 1433 sigma. Okay, so we talk about some of the 1433 zeta, 1433 tau, 1433 beta, and those kind of isoforms. So sigma is quite unique in several ways. First thing, it is the only isoform induced by PBD3. Okay? So the other isoform was not a PBD3 target. The other isoform do not have the capability to regulate MDM2 or PBD3, like sigma. Instead, they sort of enhance MDM2 activity or do the opposite. In the other isoforms regulation in cancer, actually quite a few of them, they are overexpressed in cancer. For example, zeta has been known to overexpress in breast cancer as well. So their behavior is still different in many aspects. Yeah. Okay. Any other questions? Okay. One in the back.
I have two questions. The first is there are some sounds that in the body there are many cells that do not divide during their lifetime. For example, the neuron or the oocyte. So what happened inside those cells? I mean, is there a very hard break? Certainly, is the suppressor protein that overexpressed in those cells. And the second question is there any feedback mechanism in the regulation of the cells? For example, when the DNA is damaged, protein A will regulate the protein B and C. In the case that lost the protein B, so what happened in that signal pathway? There is any aggregation of protein A?
Okay. You asked a very interesting question about the like a neuronal cell. Some of these cells already usually they don't divide anymore. The reason they don't divide is that they already withdraw from cell cycle. So meaning that they withdraw from cell cycle, already commit differentiation. Okay. Once they committed differentiation, it's sort of there's no return. Okay. And it's very difficult for them to come back to divide again. So your question about whether any break protein is elevated during this process, indeed it is. P27 or PBD7 is known to elevate it when the cell committed, like neuronal cells committed differentiation. So the break stops them from coming back to redivide again. Okay. So for the neuronal cell, these are indeed the case. For muscle cell, some of the muscle cell, when they become differentiated, they couldn't redivide again. They have accumulation of these cell cycle break protein, such as P57 or P27. Okay. As for the second question you have about the DNA damage response, if certain protein got blocked, is there any feedback? This is a very general question. I need to come up with an example to tell you maybe that's the case. For example, say we have ATM minus. Okay? ATM minus cell, they causing all the downregulation protein being compromised. The phosphorylation got compromised. Is there any feedback regulation to compensate the ATM? So far, for ATM cases, that's not the case because the ATM has been found as a human genetic disease. That there's no other way to compensate the ATM at this moment if the ATM has mutation. But other protein, we need to come up with some other example to see there's some feedback regulation happen. Okay, like I said, I encourage you to always think deep. Okay, in a scientist, think deeper. Okay, even it's kind of at the beginning, it's kind of absurd idea. Okay, some kind of not realistic, but these non-realistic ideas, sometimes you could make it happen, okay? So there's no limitation for your imagination. So you should have your creativity. Creativity based on some of the knowledge you have, and then you have some novel idea, and then try to prove these hypotheses correct or not. So when we do experiment, when we do research, there's one thing you should always remember. Hypothesis driven. Okay, what's your hypothesis? Okay, when you ask certain question, what's your hypothesis, and then how you gonna prove it? Okay, without hypothesis, you do experiments, no go. You basically based on nothing to do the experiment. So that's not good. Okay, so you need to have a hypothesis. So the hypothesis driven is very important for being a scientist when you try to do research. Okay, you hypothesize maybe this chemical compound, when you eat it, will become carcinogen, and what do they do to the cell? Maybe you could check the, hey, if I feed this kind of cell with this carcinogen, do I find the reactive oxygen species stuff, appear? Do I find the genome integrity got disturbed? Those kind of things. Okay, so you have a hypothesis, and then you do experiment to prove it. Okay. All right. Any other question? Yes, question here.
I have a question about the in vitro test that you mentioned many in your slide.
Yeah.
To prove that the role of the protein in the cell cycle signaling. But I wonder that in the body, the cell might contact to another cell, and they will receive many signaling from other cell, and they receive the signal from the protein outside the cell.
Mm-hmm.
So I think in the in vitro we can prove that the role of the protein, but in the body, the interaction between the protein inside and outside the cell may complexity. So I don't know how we prove if in the body the interaction of the protein is the same and the cell cycle in our body is the same in vitro.
Good. That is a question that I think the powerful chemistry come along. Okay? The biochemists allow us to dissect the window, the function. Okay, but in a biological context, okay, these we just only provide certain information. Like you say, so for example, we say the ubiquitination being regulated. We have E1, E2, ATP, we have the substrate, we have the E3 ligase, okay? So this is only a biochemical reaction to prove this E3 ligase indeed is causing the ubiquitination process of this certain substrate. But in the biological context, in the whole cell, we don't know this E3 ligase, whether there's an upstream being regulated, they need another cofactor to do the job much more efficient, because the biochemical reaction only allow us to dissect that window only. If we find that there is another factor, maybe we will be able to add in there, and then to say, if I boost up the reaction even 10 times better than the previous one, then you will know the whole in the biological context, the original component is only essential, but not enough to boost up further. Okay? So, I mean, this is always a biochemist we need to face. But the biochemical reaction allow us to really define the system, how it happened. But in the biological context, you certainly we need to think deep. But these we're giving time when researchers have more capability to identify more other regulators involved in the whole process. But I really encourage you to think like that, because the biochemical reaction has certain power. Inside the cell, the whole regulatory cell, like you say, is very complicated, and then really rely on A scientists, B scientists, C scientists to give you certain information, and you try to prove it, okay, in a biochemical way. Yeah. Okay. Any other question? Question over there. Okay. Okay. Either one. You be patient.
Okay, hi, sure. I have two questions for you. First, I got questions about the isoform. Is this the isomer or the isozyme?
Okay. The isoform of 14 sigma, you mean? Okay. Whether this guy is the isoform, this is not the isozyme.
So, what is the isoform? I didn't understand it.
So the isoform, okay? Isoform by meaning that, well, we should say they are homologous to each other, okay? And the isoform, we give it a name because they all have very similarity in terms of the sequence, okay? In terms of the protein organization, the motif domain organization. And then so 1433 all have a very basic capability is to form, to be able to bind the phosphoryl binding protein. So these all have a very similar one. So we call it isoform because they have similar biological activity, but they don't have the same sequence. Okay? Isozyme. What do we call isozyme? How could I come up with an example? Anybody know the answer of isozyme? Jim?
Well, isozymes catalyze the same reaction, and it can be from the same gene, or it can be from, but with alternative splicing, but it can also be from different genes with homology. Like for example, in the 14-3 family, when you have the homology of the various domains, but the function of each member of the family may be different. For example, in terms of the substrates that are regulated by the individual 14-3 members, each member of the 14-3 family regulates a different subset of proteins. Whereas if you're talking about an isozyme, all the isozymes catalyze the same reaction. For example, like creatine kinase, or creatine phosphokinase, or CPK. The different isozymes from the skeletal muscle versus the isozyme from the cardiac muscle, they all catalyze the same reaction. Okay.
All right. Any other questions?
I have one question.
Okay, sorry.
In the table, you said that the effects of some compounds on the ATM, right? You use the IC50 to measure the power of the effects of these compounds on the ATM.
Right.
In my opinion, IC50 is some kind of such unit to measure the power of the antioxidant activity. So why do we use it to measure the effects of some compounds on the agent?
You say you think IC50 is using for measure the
Antioxidant.
antioxidant. Antioxidant? You mean antioxidant? No, not just limited to antioxidant. In this case, this is the compound concentration. So the IC inhibitory concentration for 50 means 50%. Mean able to inhibit that particular enzyme or protein activity when the 50% of protein activity or the target protein or target enzyme is inhibited for 50%. So that concentration is defined as IC50. Okay? So certain drug we use it in the cancer cell chemotherapy, for example, Taxol or even some other inhibitor like Herceptin. Okay, Herceptin is the monoclonal antibody. When they were able to inhibit the cell growth from original 100% growth to 50% growth, in that concentration of the protein concentration, all the drug concentration we define as IC50. Okay.
Yes, I have a question about protein sigma.
Yeah.
Yes, I know it have an important function in transcription and translation.
Right.
So I want to ask you, what is differences between its activity in the normal cell, cancer cell, and stem cell, and what is directly change these differences?
I couldn't follow the last one. What is the what? The last part of your question.
The last part is that what is trigger these difference directly?
Okay. If I could follow correctly, what's the 1433 sigma's expression in normal cell, in cancer cell, in cancer stem cell, or stem cell, right? Okay. So 1433 sigma or 1433 protein. Are you particularly asked about sigma? Okay. So sigma's expression in normal actually is quite abundant already. But this abundant cannot be just using it as a normal guideline. There's a threshold. We talk about threshold, certain protein level had to be reached. And in the tumor cell, some of the cells totally lost the expression of sigma due to the promoter methylation. Okay? Some of the cancer cells still have sigma expression. Okay? When you do the Western blot, you still see the expression. Okay? So you cannot, based on this, just say, oh, this cancer cell still have a certain amount of 1433 sigma. Does it still contain the 1433 activity or not? We're not sure. We need to do detailed study. Okay. As for the cancer stem cell or stem cell, we have not done any study about the sigma expression in stem cell yet, so we have no idea so far. And that's a good suggestion. We may be able to explore in that field. Did I answer all your questions? Do you have other questions? The last part. Is that all? Answer your question? No? Well, so what other question do you have?
No, I don't have any question.
You don't have any question? Okay.
Hi, Maurice.
Yes.
I might miss some points in your last display lecture, but I still have not found in the internet yet that how many times in the cell cycle that the DNA would be checked for damages and when?
Okay. How many times during the cell cycle the cell will check the damage? Okay. The cell has several sensors, like I said. The mitotic checkpoint, okay, that is for mitotic process. For the DNA damaging, as we said, the sensor coming from the ATM and the mRNA complex. When the DNA damage is there, the conformation change of the DNA due to the double-strand break, this conformation somehow sends a signal to the protein surrounding these genomic DNA, such as ATM, such as the mRNA complex. And they will be able to recruit there, especially ATM because they sensing the conformation change. So original